Hello. Okay. Hi, everyone. Welcome back to this session on learning. So first we have a talk from Blaise, Aguera, y Arcas from Google Research. And as a reminder, the questions will be taken at the two microphones in the aisles. So at the end of the talk, if you have questions, please do start to line up.
All right. Shall I begin?
Yep, please go.
All right. So a huge thank you to the organizers for inviting me to talk about random big picture stuff, which is what I am going to do. The title of this talk is about as over the top as I think it could be, but I am going to make an earnest attempt to answer this question in the next 45 minutes. What is intelligence? So this is zooming way out from locus coeruleus and dopaminergic systems and the kinds of things that we've been looking at so far this morning. But my hope is that this big picture can help us to think a little bit about where we are in the whole sort of neuro-AI trajectory, and can help us think a little bit more generally about the mission, if you like. Okay, so. The inspiration for that title, as many of you will probably recognize, is the 1944 book by Erwin Schrödinger, What is Life?, which was quite inspirational to a lot of people, myself included, who started off in physics and then shifted into biology and neuroscience. And the question that Schrödinger was really trying to get at is, how is it that something that arises in the universe and that is governed by the laws of physics can appear to reverse the second law of thermodynamics, and that being the most obvious thing about life, that it seems to create order in a world that otherwise increases its disorder over time. So it's a very cool book. Anybody who hasn't read it, it's very short and it's well worth your time. And he predicted pretty much everything there is to say about DNA a number of years before it was discovered. So. The case that I'd like to make is that life and intelligence are, in fact, the same thing. Life is intelligence, intelligence is life. Also, that autoregressive prediction is basically what those things are. That is what life is, that is what intelligence is. That learning and inference are interchangeable, are also not separate. And also that intelligence has a social and self-similar and interaction-centric quality, meaning that the sort of hierarchy of scales that we tend to see in which smaller life forms band together to make bigger life forms, cells into organisms and so on, is not a just so story of how life has evolved here on Earth, but it's actually the defining property of how intelligence works. So, first of all, I will lay my cards on the table with respect to AGI, artificial general intelligence. I know this is a very contested term, but it is my belief and the belief of Peter Norvig that I wrote this essay in Neuma with in October of this past year, that we already have artificial general intelligence, that it's already here. And what I mean by that is that back in the old days when we talked about AI. The idea was never that AI was perfectly human or superhuman or super intelligent or could do everything, but rather that it was general. That a general intelligence is something that you would be able to have chit chat with, and that wouldn't be restricted to doing one particular kind of task. And what happened sometime in the 2000s and 2010s, as we began to crack all kinds of specialized problems using neural nets that had been intractable before, like speech recognition and OCR and all that kind of stuff, is that those things began getting called AI, and they're not AI, right? I mean, you can't have like a chess playing neural net and then ask it, like, by the way, how are you feeling today? Or what's your favorite Greek island? You can't even tell it, if you're playing chess, okay, let's now have all of the pawns able to go backwards as well as forwards, right? So it's extremely narrow. And that's why the terms AGI and artificial narrow intelligence, A and I, got coined in order to distinguish those narrow uses of artificial intelligence from what we actually had in the 2010s and 2000s. So, if you go to Google search and you search for definitions of artificial general intelligence prior to the year 2020, you will discover that the large sequence models that we have today, that ChatGPT and Gemini and all of that, satisfy the original definitions of artificial general intelligence. They're not narrow. They're not specific to playing chess or any other thing. I just saw a paper a few days ago about a chatbot achieving grandmaster status at chess. And that's not because it's a specialized chess system, it's because chess appears to be an instance of the broad class of problems that those large models can perform, and as such, I think that we have AGI. So that's kind of my take. Now, it's a big surprise that achieving AGI in that sense happened through basically autocomplete. And I was as surprised by this as anybody. So, my own team at Google was responsible for making Google's next word predictor for the keyboard, for Gboard. It's that little thing that shows next words on the strip above the keyboard. That is a autoregressive text predictor. It was device-sized so that the data doesn't have to leave the device, and it worked okay. Okay enough that you could save a little bit of time typing. But we knew that a system like that would never be able to achieve artificial general intelligence. It would surely require some magical pixie dust in addition to basically just a big RNN, because surely an RNN wouldn't be able to solve word problems. The next word is an AI complete problem. The next word could be Humpty and then Dumpty, and that's trivial. Or the next word could be the answer to an extended word problem, and then it would require calculating some complex word problem. Or it could be a story, and the next word could involve applying theory of mind to one of the actors or characters in the story. So there's nothing that doesn't fit within the realm of next word prediction from an AI perspective. So we knew that next word prediction using a simple neural net would never work. But lo and behold, it did. So the big shock was that by just scaling these things up and using attention, using transformers, all of those things became possible. So one can conclude two things. Either one can conclude that something can somehow pretend to be intelligent without being intelligent, or one can conclude that that is actually what intelligence is. And for me, Occam's razor says the second, and raises a whole series of interesting questions. Again, large sequence models, certainly not human level at everything, certainly not reliable yet at various stuff, but also it's very, very hard to find any more categories of problem or types of thing that they just cannot do. Whether the level of performance is below human or above human varies, of course. So the change from the narrow kind of artificial intelligence of the 2010s to the general artificial intelligence that we have today was brought about by two things. The first is the switch from supervised to unsupervised learning. If you have a supervised learner, of course, the best that it could possibly do, the most that it can do, is to score 100% on your training data, meaning reproduce exactly what you put in. That is not the case for an unsupervised sequence predictor. It's open-ended. And in particular, one of the reasons that it's open-ended is because of in-context learning, which is a property that has really been sort of dissected and figured out just in the past year and a half. I don't really have time to go into in-context learning in any detail here, but it's very, very interesting. And a kind of paradigmatic example of how that works is, if you take a large language model and you say to it something like an equiantonym is a word with the same number of letters as its opposite, give me a few examples, it will do it. And that's despite the fact that there is no such thing as an equiantonym. It wasn't anywhere in the pre-training data. And what that means is that one-shot, or in that case even zero-shot learning, is possible. Now, in a normal transformer model, that learning is forgotten as soon as the context scrolls out of view, so it doesn't stick. It's a little bit like Henry Molaison, HM, where it's a kind of Groundhog Day. Every time the context is flushed. Nonetheless, that is learning, and we now understand why that is in a much better way than we used to, thanks to the work of Johannes von Oswald, who might be here, and some of his collaborators. But basically, the attention layers can turn out to actually implement gradient descent in the contents of the context window and apply them to what follows, which is a super interesting result. And so supervised, unsupervised was a big deal. In-context learning, I think, is key to this concept of generality, because that makes the set of things that such a model can do open-ended as opposed to constrained by whatever it is that it was trained with. And also the fact that we began to apply this to language is really important, because language you can think of as kind of our Umwelt compression scheme. It's our mechanism for being able to represent to each other all of the things that we care about, and for being able to model each other's internal states. It supports theory of mind. It's a huge boost to theory of mind, and it's what allows us to band together as societies to make larger intelligences. As such, I think that language is an illustration of something that goes on much more generally when you bring smaller intelligences together to make larger ones. And it's of course what we've used for teaching and learning, and thereby becoming something much greater than our other sort of primate cousins, which are basically roughly as intelligent as us individually, but don't come anywhere close as societies, because they don't have this cultural accumulation. All right, so transformers. The transformer was the other key change. I don't think that there is anything magical about the transformer model in particular, but there are a couple of properties that it has that an ordinary RNN doesn't. The first is that it's able to implement a general Turing machine, and that's why it's able to learn things like gradient descent. So gradient descent is an example of an algorithm. They've been shown to be able to implement, any other kind of arbitrary algorithm as well, both theoretically and in practice. And that sort of brings it into a different computational class from something like an RNN. And the thing that gives it that is the multiplicative interaction of neurons in the attention layers. So that multiplicative interaction is probably really important. I don't know what the neuroscience is of that, and I think nobody knows. There are a few interesting ideas. There might be dendritic computations that look a little bit like multiplicative interactions. It might be a thalamocortical circuit. There are a variety of theories. I see this as a little bit similar to the quest for how backpropagation works in the brain, or its equivalent, right? We don't know. There are a bunch of possible ideas. But one way or another, it is likely that we have something like bilinearity or multiplicative interactions in neurons that support the same basic leveling up from simple RNNs with only linear interactions to this kind of stuff. That's not to say that what we call attention in neuroscience is the same thing, of course, as attention in a transformer, but it's that primitive, really. Still to do in transformer land and in large models is the continuous fusion of all modalities. So, we already have started to see, of course, in the last year, multimodal models that can do images and video, the very impressive Sora demo from OpenAI. And we'll continue to see continuous modality fusion be a big area. I expect robotics are going to be a really big deal over the next few years as the modalities begin to include servos and continuous motors and sensors and things. Also, making the context window infinite, aka making these models able to do continuous learning, is going to be a really big deal. So when they stop being HM-like and start to be able to bake in that in-context learning that they already do into the weights, or somehow persist that for an infinite amount of time, then they will be able to really kind of individuate and develop skills and relationships and so on, and that will be a really big deal relative to the more generic models that we have today. Third thing is that having an infinite context window will allow for active or causal learning versus the foie gras method that we use today, where you take a bunch of tokens from the internet and grind them up and force feed in random order, which is obviously not a method that is able to support things like experimentation in order to learn, right? The kind of stuff that the kids learn in Montessori schools. And my guess is that kind of active causal learning probably will lead to much greater efficiency in data than the foie gras method. And finally, we started to see a lot of mixtures of experts or dynamic ensembling of these models in the models in production. Of course, the companies have started to be a bit more secret about exactly what is behind the screen. But one way or another, mixtures of experts are important. And I think one of the reasons for that is that if you just have one transformer that is running, basically you're seeing its entire state in front of you. And we have an inside voice as well as an outside voice, and our various cortical columns or brain areas or whatever they are in there are able to talk to each other and critique each other and do various kinds of modeling before you open your mouth to say something, thank goodness. And that inside voice is probably really important in terms of being able to reason things out, critique them, look at them from various points of view, and that's exactly the kind of stuff that we're seeing happen with large models now. I suspect that a lot of the hallucination, ungroundedness, et cetera, is probably going to be resolved by techniques like that. So, this active inference or prediction idea is very old, as I'm sure many of you know. Helmholtz was a proponent of it. Also, in a little bit more modern times, Wiener and Rosenbluth and Bigelow in their 1943 paper, Behavior, Purpose, and Teleology, which is a wonderful, three-page paper, describes in detail the predictive or active inference hypothesis. Talks about behavior as being active or non-active, purposeful or non-purposeful, having teleological or feedback-based methods, being predictive versus non-predictive, and within predictive, expanding in higher and higher orders. And of course, it's exactly that predictive, expanded in higher and higher orders that we're doing today with these large, unsupervised, high-order sequence models. So, why is prediction so important? Now, I may be really laboring the obvious, but let me labor the obvious. Without predicting, you can't survive as life. So this is what I mean when I say intelligence and life are the same thing. So if you have an unsupervised model, P of X, of what you experience, and Y, your outputs, your actions, which you think of as part of the same stream, and some internal state that you learn, H, that probability distribution, learning that probability distribution is what it takes in order to stay alive. You have to predict yourself in the world, and you have to predict your own future into existence. So you can sort of demo that very easily, and I did that in this little experiment from a number of years ago with artificial bacteria. So these guys have this model, P of X action, and in this case they actually don't even have an internal state, so they lack that. But basically they start out with random such models, and the only ones that survive are the ones that don't run out of energy. And they learn how to do chemotaxis on the basis of that. Because if you don't predict your own survival, then, well, let's see. If you do predict your own survival and your model is a good one, then you will live to predict another day. If your model is not very good and you can't effectively predict your own survival, then you also will not live to see another day. So the only scenario in which you will live to predict more is if your prediction involved your own future prediction and its continued existence, right? So inherently, a predictive model of sequences in which you are playing a part, that is to say, in which your own actions are part of that sequence, will satisfy the survival imperative, and that's really kind of what it boils down to. There are a lot of ways of skinning that cat, of course. These are multiple different populations of these bacteria that learn different kinds of behaviors that are all consistent with survival. And if you start to give them the ability to signal, then they start to behave, together symbiotically and have all kinds of interesting behaviors. So, you can kind of play this game at higher and higher orders. So, how does this game work at higher orders? How does multicellularity arise, given that sort of very basic idea of active inference? Well, if you have two cells and they predict each other, that is to say, each one's actions include the other's observations, and they start to have internal states, then there are conditions under which they will form a symbiotic relationship, meaning that their joint survival will be higher if they are effectively able to predict each other as well. And voila, that's how multicellularity arises. I'll give you an example. So we all know the story of Hydra, So this is a case in which, this is how nerve nets first arise. We start off with muscles. So it's a muscles first world. It's not a neurons first world. And the first neurons arise in order to allow the muscles to work in better synchrony. So I'm sure many of you have seen like Strogatz's old work on synchronization of fireflies. They form this kind of strobing. They do this beautiful dynamic synchronization. That, of course, is mutual prediction, right? Each firefly is trying to predict when the other ones are going to light up and light up at the same time. It's the simplest form of prediction. The muscles in a hydra or a jellyfish have to do the same thing if they want to actually work together in order to swim. So each of them will want to predict what the other ones are doing. Now, if they only can communicate with their neighbors, then any kind of prediction is going to be really hampered because it's going to be limited to the speed of light, right? To the speed of those traveling waves that can go around the animal, and that's slow. So what will each muscle try to do? Well, it will try to grow sensors that will sense other muscles farther away than the neighbors. And that's the way I think about the nerve net of a hydra. It's not that there is a distributed brain. Rather, it's that each muscle cell has grown a network of sensors in other muscle cells that will help it to predict at longer range its further away cousins. So what happens now when you start to have something like an Acella, one of these little kind of very primitive flatworms? Well, if it's moving along in a certain direction in its benthic environment, and it can, say, both move forward and back up, and there are two different gears now, not just a single gear of move forward, then every muscle is not only going to want to know what some of the other neighboring muscles are doing, but is going to want to also tap into the sensors at the leading edge of the organism, because those sensors are going to have the most information about whether going forward or backward is a better idea. So they're all going to grow their sensory neurons to preferentially end up at the head. And that accumulation of sensors at the head is, of course, the baby brain. And these are, all these cells are themselves reproducers. So as they start to reproduce in the head and be able to predict over longer timescales, that's also a form of symbiosis, which will then make what the muscles are tapping into for the brain work better. I really like this picture because it's kind of backwards, right? So we usually think about in a sort of homuncular way about the brain driving the body, but in this picture it's the body that is driving the brain. And that's just back propagation, of course, right? Forward propagation in a neural net only exists because it has been learned by back propagation, and back propagation always works from the muscles on back. So it's your muscles that are driving your brain and that have driven the exigency of your brain to predict at longer and longer timescales. So as you start to get cells proliferating in the head, those brain regions or those multiple cells are going to want to start to predict each other as well. The long-range connections in cortex you could think about as one brain region growing sensors into other brain regions in order to do a better job of predicting itself and predicting its neighbor. That has some pretty interesting implications if you start to think about it beyond the individual as well, because it implies, for instance, that our interpreter, our left hemisphere language region, is really an outpost in your brain on the behalf of other people. In other words, the idea that when you talk, you're the one doing the talking, and you're the volitional one, maybe is backwards. Maybe it's more the case that the communicative act is actually driven by the receiver. If you think about it, if you think that you've been successful at a communicative act, but the receiver doesn't think you've been successful, well, the communication has not been successful. On the other hand, if you think that you haven't communicated anything, but your receiver thinks, has learned something, then it has been successful, and that will get reinforced. How would we test a hypothesis like that? Well, if there are communicative acts that are involuntary, then that's exactly the case where you've gotten some things reinforced that aren't necessarily ones that you would have wished. That's what blushing is. That's what the Duchenne smile is. That's what sweating is. When you're having a hard time in an interview or whatever. And all of those are communicative acts that look like outposts of your recipient's brain and that have actually wired up your own brain and are doing back propagation through language or through communication out in the environment. So yeah, that's how I see it. This also leads to a relational picture in which you don't necessarily think about stuff like consciousness or entityhood as a property that a particular brain has or a piece of matter has, but it's something that is more fractal and is really based on those relationships. between signal and motor and whatever it is that will be informative for that motor stuff to act. So, it's relationships all the way down. And this has some pretty interesting implications for things like blind sight and split brain experiments. I don't think I have long enough to go into these in any detail, but to give you a blind sight example, when Nicholas Humphrey and colleagues discovered blind sight way back in the day, one of Humphrey's conclusions is that since we're not able to report anything that we see with our subcortical vision pathway, it must be the case that the cortex is the conscious part, and that the subcortical part is not conscious, and therefore that you can separate out competence and awareness. Well, there's another way of looking at it, of course, which is that everybody in your brain is modeling everybody else. It so happens that the interpreter, the thing that you can talk to, doesn't have access anymore. Is not wired up to or talking to that subcortical vision pathway anymore. This doesn't imply anything about the consciousness or lack of consciousness of the subcortical vision pathway. It's doing its own modeling. It's just that, as with a split brain patient, those two systems are now isolated from each other. So this relational picture leads to some pretty interesting stuff. It sort of leads to the idea, for instance, that from the outside a person can look like one entity. From the inside, there can be multiple relationships there that sort of, there's something pretend, right, about the idea of unity of consciousness. Okay, so let me bring things a little more down to earth. Here's another really great piece of classic work. It was the last thing that von Neumann did before he died, and this theory of self-reproducing automata was finished by Arthur Bergson and published posthumously. Von Neumann also predicted DNA, and he did it by extending Turing's work on computation to the idea of reproduction. So he basically said computing is needed in order to reproduce. So we've talked about how computing is needed in order to predict your own future survival. Computing is also needed in order to reproduce your body, and the reason is that Neumann's sketch of what reproduction looks like is, well, in order for something to build something of equal complexity or even greater complexity than itself, it has to be the case that there is a tape that specifies the instructions for doing that building. And so, he predicts the DNA in this. And not only is there a tape, but there has to be a machine that reads from the tape and puts pieces together to make things according to the instructions on the tape. That would be the ribosomes, of course. And furthermore, there has to be a machine B that copies the tape. So if the tape includes instructions for building machine A and machine B, now you have a self-reproducing system. This is all a generalization of Turing's general theory of computation, but with sort of, with hardware involved now. And as you will appreciate, of course, all this is exactly correct. This is DNA polymerase and ribosomes. And machine A and machine B, by the way, both need to compute. They both need loops, they both need branches, and they both need to be able to execute instructions. So, this is a strict generalization of computing per Turing. So I'm going to show you now a few little experiments that I sort of played with, back at the end of last year, which I called BFF, which you will find out what that stands for shortly. But this was an exploration of this idea of computing for reproduction and some of its implications. So, brainfuck is an esoteric programming language created in 1993 by a German computer scientist, Urban Müller. It is a kind of minimal Turing machine. It has only 8 instructions. This is the entire instruction set that you see on the screen in front of you. In its original form, it has 2 tapes. There is an instruction tape and a data tape. And there's a console as well. So the instructions are increment the data pointer by 1 on the data tape, decrement the data pointer by 1. The data pointers generally begin at the first position. Increment the value at the data pointer by 1, decrement the value at the data pointer by 1. Output the byte of the data pointer or input from the console to the data pointer. And finally, these open bracket, close bracket instructions, which give you looping. So at open bracket, if the byte of the data pointer is 0, jump to the matching close bracket. Otherwise continue. At the close bracket, if the byte of the data pointer is non-zero, jump back to the open bracket, otherwise continue. That's it. And with that, you can write Microsoft Windows or whatever you want on although it will not be very easy to debug. This code will print hello world, I'm told. I did try it. It does work, but God help me if I can figure out how. So here's how I went from BF to BFF, and you can guess what the second F stands for. So I made it self-modifying, so rather than having two tapes, one for instructions and one for data, there's only one tape. That tape is both instructions and data. The data pointer begins at some position in here, and the instruction pointer begins at the first byte. And the console is also the data, so there's no separate console either. So there's a console pointer and a data pointer, and the first 2 bytes specify the initial value of the data pointer and of the console pointer. And then the instruction pointer begins on byte number 3. I'm using very short programs, so just of length 64, and you start off with a soup or a stew that has 8,192 of these 64 length programs in them, in the stew, and they begin all random, so just random bytes. Now, when it's just random bytes, that means that only 1 in 32 of those bytes is going to even be an instruction. Anything that is not an instruction, by the way, acts like a no-op, so it just advances to the next byte. Now, the procedure is you pluck 2 of those strings out of the soup at random, you stick them end-to-end, concatenate, and run. And then you take them back apart, and you put them back in the stew, and that's it. All right, so just do this over and over, put them together, run, take them apart, put them back, and keep going. And what happens? All right, I will show you what happens. The way I'll show you is with an old school console video. The numbers on the left, the 11111 is a histogram, so that's a count of all of those strings. Of course, they're all unique to begin because they're all random. You'd expect them all to be unique. And you can see ops per interaction over there. So there are only 2 ops on average, 2 operations being run per interaction, as you would expect, because if only 1 in 32 of these bytes even codes for anything, then it's going to do very, very little computation initially. All right, let's watch it run. 1 million, 2 million, 3 million, 4 million, 5 million. And something is happening. And bam, we have life. So I think this is pretty cool, because you go from pure randomness to something that looks like it has taught itself how to program. Better than I can in brainfuck, for sure. And these things, what are they doing? They're obviously reproducing. You can see that because the counts, the count of the most common one is 5,000, and then 297, and 99, and so on. So these strings are reproducing. They've learned how to reproduce. And also they are computing like nobody's business, right? So we went from 2 operations per interaction to 4,800 operations per interaction. So we went from no computing happening to a lot of computing happening. Okay, and this is what that looks like. That phase transition looks like in sort of computational terms. So there are 10 million dots on the screen, 1 dot for every interaction, and the vertical position of the dot is the number of operations that ran in that interaction. And in this particular run, you can see that right at about 6 million interactions, suddenly there is this dramatic phase transition where things go from not computing to computing. Here's another really nice way to visualize what's going on. So in red is the Kolmogorov complexity of the soup. So this is, it's an estimated version of the Kolmogorov complexity by just zipping the soup. So you run LZMA and you take the ratio to the original size. And you can see that it's a gas in the first part. The ratio is 1, it's incompressible, it's just noise. And at that transition, we suddenly get a transition to order. Now, it's not a transition to crystalline order. Crystalline order would be there's only 1 reproducer and there's just the occasional noise. It's not that, right? So it's considerably non-zero amount of information, but it's much lower than the noise. So when that transition happens varies depending on the random number seed on the run, but it always happens, and it happens to a stereotyped level of complexity. So why does it happen? Well, it happens because replication is more stable than randomness. And this is an idea that was developed very nicely by the chemist emeritus, Adi Pros, in Israel. So he talks about dynamic kinetic stability in abiogenesis, in the origins of life, as a way of explaining why it is that DNA is more stable than granite. Right? So obviously, granite lasts a long time, but it's passive. It's not active, and so eventually it will succumb to the second law. How does one not succumb to the second law? Well, one doesn't succumb to the second law if you have a dynamical process rather than a static one in which you are actually reproducing yourself, and then suddenly that's the way that you push back against the second law. And what von Neumann showed is that requires computation. So as a result, computation is an attractor. And we can see that computation is an attractor by the hugely increasing amount of computation happening in the soup. Now, I should say, though, that to say that it is stable is not quite accurate, because evolution is never done. So, it's more stable than static, but it doesn't stay still. Evolution is always in progress. There's actually a cascade of phase transitions involved, which I'll show you in a moment. And the reason is that replicators create niches for other replicators, just like the reproduction of a snake creates a niche for the reproduction of a snake's ribs. Right? So, Hox genes, get like lots of reproduction of the snake's ribs. That's a reproducer as well, just like neurons in our brains are reproducers. And it is the existence of the snake that gives the niche for the ribs to reproduce, just as it's the existence of our brain that gives the neurons the niche to reproduce. So, in this picture, symbiogenesis, meaning the symbiosis of replicators at a smaller scale, enhancing the dynamic kinetic stability of the larger structure, is the key operation that leads to speciation or to development or to evolution. All right, so here's what the information picture looks like at a byte level. So this is all 64 bytes, and you can see that transition happens, right in this case, right about at 3 million interactions, and that the number of bits per byte, really goes down. Although there are certain bytes that are very highly conserved and certain bytes that are not so highly conserved, and this is. Of course, similar to what you see with DNA, where there are some things that are very highly conserved and some that are not, some that are sites of mutagenesis. And by the way, the method of mutation is very, very different from what sort of Jacques Monod imagined, where it's just random noise and selection. Right? When a snake gets an extra rib, that's not just a random mutation. Right? It's a self-modifying system. So mutations have a certain structure. And those mutations can result in very non-trivial changes in morphology or in function because the system is self-modifying, that is to say, because it's code, because it's computational. Here's what that looks like in a little bit more detail. So this is, I'm now showing a log of operations. And the transition from green to red here is that red is when you start to see replication. In other words, a string that is duplicated. And black is strings that are very, very highly duplicated. So you can think of those as species. Species that emerge at a certain point, and you see that there are ongoing sequences of phase transitions of various kinds. What do those phase transitions look like, and why are they happening? Here is something else cool to sort of give you a little bit of a sense. So this is the number of coding bytes in the string. Now remember, the default is 2, right? 1 in 32 is randomness. You can make a replicator with 5 bytes in this brainfuck language, but you can see that the number of coding bytes continues to increase, right? Even after that phase transition, it increases and increases. In this case, plateaus at about half of the bytes. So why does the computation continue to increase, right? The number of operations continue to increase even after the phase transition? Well, you can get a bit of a sense by looking at plus L replications, meaning replications that involve the entire first string copying itself, or some part of it copying itself all the way to the other half. That's those guys to the right. And you can see that they begin right before the phase transition, unsurprisingly. That's when you get this exponential growth of those replicators. But something happens before that that looks like inexact replication. So these are replicators of some chunk of code that don't necessarily copy the entire string, but just copy some substring and are not aligned to the length of the whole thing. And you can see they begin significantly before the phase transition. So that's rightward replicators. That's leftward replicators, which are a little less common. So those rightward and leftward imperfect replicators create the conditions for the evolution to continue on to the full string replication. And then once the full string replication happens, you start to see the emergence of multiple replicators. So a multiple replicator is when you have a piece of code that copies itself more than once in a single run, like a snake's rib. So as you see, multiple copies show up in the target. And of course, the moment you have something that copies itself multiple times, it has to have conditional behaviors, because now it'll get run twice after it's copied, and it needs to behave differently under those two different conditions. So it starts to have an ecological niche in which it has to behave differentially. And you can see how this begins to connect to prediction, right? I mean, is it a predictive model? Well, I guess that's a matter of what our model is of a predictive model. And they're not having a detailed theory of mind about each other yet, so we can't really ask them. But I'm guessing that if we wait a few billion years, they probably will have that. Okay, so I think there's a very intimate relationship between prediction and reproduction. They both require computation. They both involve a model of the self, whether that's your DNA strand or your theory of mind. They both create a dynamical system which underwrites dynamic kinetic stability, and they both push back against the second law, and this is why I say that I think life and intelligence are the same thing. This is that kind of identity, at least in words. So computational symbiogenesis is I guess, another fancier way of how I would define this. And this whole thing is fractal, meaning that if an agent's Umwelt is other agents, which are themselves predictors, and larger intelligences are made out of smaller symbiotic intelligences mutually predicting each other.
Sorry,
Stop.
Just in the next couple minutes.
Yes, I think I'm at my second to last slide. So there are going to be scaling laws that allow selves composed of larger populations of mutual predictors to form larger social units that can themselves be more intelligent. And this is exactly what we see in the work of Nicholas Humphrey and Robin Dunbar with the social brain or social intelligence hypothesis, that as brains grow among primates and among dolphins and so on, troop size grows too. So there's a parallel, in other words, between how many cortical columns you've got and how many minds you can model in your troop as well. And this kind of shows you how the two levels of this fractal are coupled together. More intelligence at the smaller level leads to greater intelligence at the larger level as well. So there are a lot of implications to this way of looking at things, which maybe I will leave to discussion and end there. Thank you all. Did I run too badly over? Do we have time for a couple of questions?
Yeah, so let's take a couple questions, but can we have the next speaker start to come up? And remember to please come up to the microphones in the aisles as fast as possible.
Hello, hi. So when you say survival needs prediction, I think you're talking about something quite similar to Conant and Ashby's good regulator theorem. That leads to two challenges, as I see. One is, how do you arrive at good models, and how do you use the good models to create good decisions? So how do you use, for the first one, how do you use all of the dynamic kinetic stability to arrive at a good models for large language models? And when it comes to planning, even something as simple as MCTS is not computationally feasible. So do you even need optimal decision making, or is good enough decisions good enough?
Yeah, I don't think that you need anything like optimal decision making. The idea that life or intelligence are optimization, I think, is not quite right, in the sense that optimal for what, right? Is always the question. Like in a lot of the classic work on cybernetics, there was something external that was being optimized, and where I think that cybernetics needs the update is that it's each other, right? It's predictors predicting each other. So I think it's the social aspect of it is at the core of what I think you're getting at. It's a longer conversation, obviously.
Thank you.
All right, one more on this side.
If I may, now that I have the microphone, I'm going to go ahead and ask 2 questions. First, thank you so much for the...
You'll ask one question, thank you.
Okay. Thank you for the inspiring talk. So I'm going to go ahead with my first question. So when you were talking about the multi-cell organisms, you talked about prediction as kind of the goal of life. I couldn't stop wondering how this could be the ultimate goal, because I keep thinking about the case that maybe if you have multiple cells and now your cells start to die, the prediction becomes super easy and super accurate. So the cell keeps predicting everyone's dead, everyone's dead, everyone's dead. So how is this going to help with survival?
Right. So that's a great question. The sort of the name for that is the dark room problem. Right. So if you predict that you will do nothing and experience nothing, and just go into a dark room, then your prediction accuracy will be perfect. What prevents that from happening? And I think the answer is symbiosis. So ultimately we all feed each other, and by going into a dark room, you are cutting off your relationships with everybody else, and you will therefore not survive. And that's true at the level of an individual. It's true at the level of a brain area too, right? If there's a brain area that has that pops up in a mutant, and it doesn't actually help the other brain areas around it, right, by providing them with better predictions, then even if its own measure of prediction is very high, it's not going to be conserved because it's costly. So I think symbiosis is the answer to that question. All right, and I'll step away. Thank you all so much.
All right, thank you very much. So next speaker is Carla Golden, who will talk about estrogenic control of reward prediction errors and reinforcement learning.
Yes, hello. Thank you to the organizers for the chance to share this work, which we recently posted as a preprint on bioRxiv. I'm a postdoc in Christine Constantinople's lab at NYU, and we found that estrogen, which is a hormone that's expressed both throughout the body and the brain, modulates learning. The most potent form of estrogen in the brain is called estradiol, which has multiple receptor types that are distributed throughout the brain, including in reward-related regions. When bound to its receptors, estradiol can regulate molecular expression, both indirectly through intracellular signaling cascades and directly as a transcription factor. However, we know very little about how estradiol, through these mechanisms, can influence cognition. And this becomes important when we think about health, because many cognitive symptoms of psychiatric disorders fluctuate in their severity across the menstrual cycle as hormones change in expression. So this suggests that those hormones, such as estradiol, could be impacting these behaviors. For example, in the follicular phase of the menstrual cycle, when estradiol peaks, stimulant use becomes rampant in individuals who are suffering from addiction. Then, when estradiol rapidly declines during this phase, there's a higher risk of ADHD symptoms. And in the luteal phase, when estradiol is relatively lower than progesterone, memory is worse than schizophrenia. So one way estradiol could be influencing these behaviors is by modulating dopaminergic signaling. Estradiol has been shown to modulate dopamine reuptake, release, receptor expression, and synthesis, but these effects have been elucidated using exogenous doses of estradiol, so we know less about how endogenous estradiol, or estradiol that's made naturally in the body, could be affecting dopaminergic signaling and what this means for cognitive behavior. We're interested in how estradiol affects reinforcement learning because it's isolated such a robust relationship between dopaminergic signaling and reward prediction error, or the difference between what was expected and what actually happened. As I'm sure many of you know, in 1997, Schultz, and Montague showed that dopaminergic neurons in the ventral tegmental area, the VTA, in the midbrain, encode reward prediction errors. They increase their firing in response to an unexpected reward and suppress their firing when an expected reward is withheld. These VTA neurons then project to the nucleus accumbens core, where they release dopamine that also encodes reward prediction error. So given the relationship between estradiol and dopamine, we wondered, how does estradiol modulate dopamine in the nucleus accumbens core during reinforcement learning? And this is the question that we address with this work. To study the effect of varying levels of endogenous estradiol on behavior, I'm using vaginal cytology to track the rat's estrus cycle, which is their reproductive cycle. There are 4 stages of the estrus cycle. Each lasts for about 12 to 48 hours, and the vaginal cells appear differently in each stage. Estradiol is low during metastrus and diestrus, which are the non-fertile stages. It peaks during proestrus into estrus, which is when the rat ovulates, and these are the fertile stages. So with vaginal cytology, I can determine the stage that the rat is in and infer the amount of estradiol that they're expressing. To study the effect of estradiol on reinforcement learning, I'm using a task that our lab created where rats work for water rewards that vary in size. The task is self-paced, so the rat decides when to initiate each trial, and they do so by making a nose poke and a center port. This triggers an auditory cue, the frequency of which indicates to them what amount of reward is going to be available on that trial, and it can be one of 5 different volumes of reward. Trial initiations are the epoch of the task that I'm most interested in because they're shaped by previous experience, which suggests that they're governed by reinforcement learning. So here we regressed initiation times against previous rewards and found that previous rewards have a negative weight on initiation times. The rats are more motivated when they receive larger rewards, so they initiate the next trials more quickly. And this effect declines as an exponential function of time, where the most recent rewards have the greatest impact, and this is a hallmark of reinforcement learning. To manipulate their expectations about reward, we vary the reward sizes across blocks of trials. In low blocks, only the smallest 3 rewards are offered. In high blocks, only the largest 3. And in mixed blocks, all rewards are available. And this is an uncued and hidden structure of the task that allows us to figure out whether we can impact their expectation of reward because we're varying reward history, and therefore we're varying the value of the state. We found that varying the value of the state does indeed impact their initiation times. Rats initiated trials at a faster pace in high blocks, presumably because they're more motivated by those larger rewards. And then they take longer to initiate in low blocks. And here we believe this is because there's a cost to initiating. So when they're getting small rewards, they're not as motivated to initiate trials because it's probably not worth the effort. In fact, across the 306 rats that have trained on our task, almost every rat's initiation times are sensitive to the value of the state in this way, where they take longer to initiate trials in low blocks. Interestingly, if we look across the estrus cycle, in proestrus and estrus, which are the fertile stages around the time that estradiol peaks, we see that they have pronounced sensitivity to the value of the state compared to when those same exact rats are in metastrus and diestrus, the low estradiol non-fertile stages. You can see that the difference between initiation times in low and high blocks here is greater in the fertile stages compared to non-fertile. And then males look more like the low estradiol stages, which makes sense because they also have lower levels of circulating estradiol and fewer estrogen receptors. So we can conclude from this that the sensitivity to the value of the state maps on to estradiol activity. Then if we look at the trial-by-trial effect of reward history on initiation times, here in mixed blocks when any reward is possible, and just in the rats that had the enhanced sensitivity in the fertile stages, we can see that the regression coefficients for the previous rewards have a more negative weight in fertile stages, suggesting that these previous rewards have a greater effect on their initiation times. So together, this suggests that there's enhanced learning during the fertile stages when estradiol peaks. To then test whether reward prediction error encoding is stronger during fertile stages and maybe driving this enhanced learning, I measured dopamine activity during the task using fiber photometry and a GrabDA sensor of dopamine in the nucleus accumbens core. We found that reward prediction error is encoded during the reward offer queue, which makes sense because this is the first time the rats are learning what amount of reward is going to be available to them on that trial, and they can compare it to their expectations about reward. We find a striking monotonic relationship between the dopamine response and reward volume, and they even have negative reward prediction errors in response to the smaller reward offer cues. But what really cinched this as the period in which reward prediction error is being encoded was that the response to the same exact reward size depended on the reward block in which it appeared. 16 microliters was available in all reward blocks, but it was better than the value of the state in low blocks and elicited a positive reward prediction error, and worse than the value of the state in high blocks and elicited a negative reward prediction error. So this response has more to do with relative value than simply reward volume. Interestingly, if we look at reward prediction error encoding across the cycle, in non-fertile stages, encoding of the reward prediction error is relatively restricted compared to fertile stages where encoding is enhanced, particularly here for the largest rewards, 32 and 64 microliters. And then if we plot the area under the curve of the phasic response as a function of reward volume for each reward block and across rats, we can see that in mixed and high blocks, reward prediction errors were significantly enhanced in fertile stages. Here are the solid curves. And this is, again, just for the largest rewards. So this suggests that there's a gain on reward prediction errors in fertile stages that could possibly be driving their enhanced learning. To test whether enhanced reward prediction errors for larger rewards could account for the observed behavioral effects, we use a reinforcement learning model that Andrew Ma, a graduate student in the lab, created that assumes that initiation times are inversely correlated with the value of the state, which is updated by reward prediction error. To model fertile stages, we added a gain on the larger reward prediction errors, depicted here in orange, and compare this to not adding a gain, depicted in purple, to model the non-fertile stages. And we found that this gain on larger reward prediction errors could indeed replicate the greater sensitivity to the value of the state and the enhanced trial-by-trial learning that we saw in the fertile stages. So this suggests that the gain on reward prediction errors in fertile stages for the larger rewards that we detected with photometry could be driving this enhanced learning. We also found that increasing dopamine release in the nucleus accumbens core using optogenetics drives down trial initiation times. So this suggests that there's a causal relationship between dopamine RPEs and initiation times. We next wondered what mechanism could underlie enhanced reward prediction error encoding in fertile stages. And as I mentioned, estradiol can regulate molecular expression. So I used mass spectrometry to detect differences in protein expression in the nucleus accumbens core across the cycle. And this is an unbiased method that allows us to ask what aspect of dopamine signaling, if any, is altered across the cycle. Interestingly, two proteins that reuptake dopamine from the extracellular space back into the presynaptic cell, which are expressed on axon terminals in the nucleus accumbens core, the dopamine and serotonin transporters, are both reduced in expression in fertile stages. So this suggests that dopamine clearance takes longer when estradiol is higher. To then determine whether this reduction in dopamine reuptake could account for the enhanced reward prediction errors that we detected, specifically for larger rewards, we simulated what a reduction in dopamine clearance would do to phasic dopamine. At the microsecond timescale, if reuptake is slow, once dopamine is released, it'll take longer to return to baseline. So this would increase the likelihood that dopamine from consecutive action potentials would temporally summate. We then estimated fluorescent signal by treating it as a noisy observation of the underlying dopamine concentration, and found that averaged over many trials, temporal summation in the slow reuptake condition is sufficient to produce the larger phasic responses for larger reward prediction errors as measured by area under the curve here. So decreased expression of dopamine transporters could be the mechanism by which reward prediction errors are enhanced for the larger rewards in fertile stages. Lastly, estradiol is one of many hormones whose levels fluctuate over the estrous cycle, so we wanted to determine whether estrogenic signaling specifically is necessary for the hormonal modulation of reinforcement learning. We hypothesized that estradiol regulates the expression of transporters in the VTA, so we knocked down estrogen receptors there. And to do this, I borrowed a virus from Takashi Yamaguchi and Dayu Lin's lab at NYU that knocks down estrogen receptor alpha by targeting its gene ESR1 with a short hairpin RNA, or shRNA. The virus is induced by doxycycline, which two excellent techs in the lab, Audrey Martin and Daljit Kaur, delivered to the rats through their food and water, so that when the rats were given dox, they no longer expressed estrogen receptors in the VTA. Here we're using rats that had enhanced sensitivity to the value of the state in fertile compared to non-fertile stages, just as I've been showing you from the beginning. We found that inhibiting estrogen receptor alpha with induction by dox suppressed sensitivity to the value of the state in fertile stages, depicted here in dark green, and made the initiation times look more like non-fertile stages. And this was true across 10 rats, so it suggests that estradiol, by way of its receptor in the VTA, is the main regulator of this behavior. It also suggests that enhanced learning in fertile stages isn't due to a systemic effect of estradiol, but specific to its actions in the midbrain. So in conclusion, our working model is that estradiol suppresses expression of dopamine reuptake proteins, which allows for dopamine to accumulate in the extracellular space, which enhances reward prediction errors, particularly for larger rewards, which in turn enhances learning from reward history. So with that, I'd like to thank the Constantinople Lab, my funding collaborators, and you all for listening. And to hear more about the temporal wagering tasks that our lab created that I only touched on very briefly, you can go to Shannon Chirac, Hee-Jae Jang, and Danilo Trinidad Perez Rivera's posters, which are coming up next. So I'll take questions. Thank you.
Could you speculate on what might be the evolutionary advantage of this sort of phenomenon?
Yes, I can only speculate because we don't have the tools to test that, but it's definitely something I'm very interested in and matters a lot. Jill Becker has shown that dopamine in females is important for regulating mating with males, where they have to time mating by hiding behind a barrier, which increases the chances of them getting pregnant. And if there's no dopamine, then they don't do as good of a job pacing. So whether dopamine there has something to do with like timing of behavior, I'm not sure, but I think it's really interesting. Yes.
Hi, beautiful work. Seems like there's also a baseline difference in terms of learning compared to comparing the non-fertile females and the males. Can you sort of elaborate on explanation for that?
Yeah, so estrogen receptors, which are really important for the estradiol activity, they vary in their distribution and their density between males and females. So I would guess that that's what it has to do with, is that there's still more estradiol activity that's happening in the non-fertile stages compared to the males.
So like a common mechanism that explains.
Yeah, potentially.
Yeah, thanks.
Let's take one more question on this side while the next speaker starts to set up.
Hi, thank you for a very clear talk. What do you expect would happen if you were to artificially create a cycle in males?
Yeah, I think, just going along the same mechanism, I think that it probably would enhance learning in males. I mean, it's something that we need to test. But they do have fewer estrogen receptors, so I don't know if the activity would really match the same levels.
Cheers.
Thanks.
Okay, thank you again. And our next speaker is Ching Fang, who will talk about predictive auxiliary objectives in deep RL, mimic learning in the brain.
All right. Hi, everyone. I'm a PhD student at Columbia, and I'm sharing this work with Kim Stachenfeld. So here we're looking at how deep reinforcement learning systems can be a good way to model representation learning in the brain. So in computational neuroscience, we're often studying regions in isolation. For instance, the hippocampus has long been studied as a region where predictive maps are computed and where you're learning the transition structure of an animal's environment. But we know that this is a simplified picture and that in reality, many regions are connected in very complicated ways. So for instance, hippocampus receives inputs from sensory cortices, which has been implicated in self-supervised representation learning. And these regions also are connected to the striatum, which has been connected to model-free reinforcement learning. And so even in this simplified anatomical picture, we can see that it's not obvious the best way to model how different computational objectives interact and how representations from one region may shape representations in another. And so what we hope to convince you of is that deep reinforcement learning offers a natural way to model the way multi-region objectives interact. So to give some background on reinforcement learning, we're assuming that the agent is in some environment where the environment provides information about reward, here whether or not you've reached a strawberry, and information about the state of the animal. So here, knowing that the animal is 1 meter south of the hedge. And with this information, the agent can take reward and state and learn Q values in the model-free setting. And so these are the expected future reward if you take some action from some state S. And by learning all of these Q values, the agent can then take the best action at any state and take that action within the environment and repeat. But in deep reinforcement learning, the problem becomes more complicated. So we can no longer take for granted that the state information of the agent is provided, but rather the agent must estimate this information itself. So here the environment may be providing high-dimensional sensory observations, say a visual scene of a park, and the agent, through an encoder network, has to learn a low-dimensional state representation Z to understand where it currently is in its environment. And this is then used to learn the Q values, this time through a deep neural network. But even in this case, representation learning is quite hard, and it's often not enough to just have a system that looks like this. In machine learning, you often need to additionally regularize the state representation learning with what are called auxiliary objectives. And these are extra loss functions that you demand on top of the state representation Z. And they're useful for many reasons. One is that they prevent overfitting to the current value function. The other is that they prevent what is called representational collapse in sparse reward environments. And so this is a failure mode where your encoder begins mapping all of the observations into a single point. And an interesting thing about these auxiliary objectives is that in ML literature, many of these objectives are actually predictive in nature. So that means they're requiring the agent to predict the next observation or the next latent state. And from a neuroscience perspective, this is very intriguing because we see prediction all throughout the brain, and particularly in the hippocampus. And so all of this guided our development of a deep reinforcement learning framework that we think can map on nicely to multiple regions of interest. So here, the encoder network is analogous to your association areas, and the model-free Q-learning system is analogous to the striatum, and a predictive network that will act as an auxiliary objective will be similar to the hippocampus. And in the simulations we run, we're going to assume the environment is providing information about reward through this scalar reward signal, and observations will be visual inputs from a 2D grid world environment. And so just for a last little bit of setup, I'll give a simplified description of what exactly our auxiliary objective is. Let's assume the predictive network is described as some function t that takes in your current state z and some action that was taken a. The way that this is trained is that we're going to look at an example transition that the animal has previously made in its experience. So here, this animal or agent has started in state Z, taken action A, and ended up in state Z prime. In this case, the loss for the predictive network will look as following. This is essentially asking that the T function is able to predict the next state if it's given the action A and the state Z that the animal has taken. Okay, so with this setup out of the way, the first thing we wanted to understand is how these predictive objectives are able to make learning more efficient. So the task that we'll look at is a 2D foraging task in Grid World. The agent is essentially tasked with navigating to some hidden reward, even though it could be initialized randomly throughout the arena. And here I'm showing the episode score, where 1 is the maximum throughout training, several training steps. And we can see that the agent, when given this extra predictive objective, is able to learn faster and also achieve maximum reward better than the agent that solely has the Q-learning loss function, which is in blue. We can also get a better sense of what exactly is happening in representation space. So here, I'll be labeling the states of the environment according to which quadrant they're from with these 4 colors, and the goal state will be labeled in red. And we can then look in 3D PCA space to see what state representations are learned by the model. So in the model without any extra objectives, we can see that the state representations tend to be expanded around the point of reward, but otherwise pushed together when they're farther from reward. Whereas if we look at the model with the predictive objective, the state representations better respect the global structure of the environment. And if you're interested, you can also see our paper for comparisons to long horizon prediction and experiments in representation transfer, which I won't go over today. But now that we have a better idea of representational effects in the model, we then want to see whether there's a correspondence with representational effects that are seen in the brain. So here I'll be looking at neurons within the predictive network and asking whether they can capture results in the hippocampus. I'll go briefly through this and just say that the model captures a lot of classic hippocampus results. For instance, the emergence of place fields, predictive skew, where place fields will tend to move backwards in the direction of movement after learning, and also reward effects, where place fields begin to pile up around the location of reward. And again, if you want to see more, we also test the model in a slightly more difficult setting where the task is partially observable and the model has to be run recurrently to solve the task. And we use this kind of task setting to show that splitter cell phenomena needs both memory and prediction to capture the full breadth of splitter cell results. But finally, the last bit I want to show is our comparisons of model representations to visual cortical data. So here I'm looking at neurons within this encoder network. This is a convolutional network with a multi-layer perceptron layer, and we're going to compare that to visual cortical data. So we did 2 sets of experiments for this. One is an experiment from Port and Kahn in 2015 that shows how value learning affects visual coding in mice visual cortex. So here the mice is running down a virtual linear track, and as a mouse runs down this track, occasionally it will enter a vertical striped corridor, which leads to reward, or it may then start running down an angled corridor, which doesn't lead to anything at all. And what the authors see is that neurons in the V1 cortex tend to become more selective across learning. So they become more selective for both vertical and angled conditions, but this boost is seen even more for vertical rewarded conditions. So that is, their selectivity seems to be biased for the rewarded corridor. And we also see a similar effect in our model where across learning, more neurons become selective for the vertical rewarded condition. And importantly, you really need a value learning module within the system to get this skewed effect. So finally, the last experiment we look at within visual cortex comparisons is this experiment from Lee and DiCarlo in 2012. So here they're recording from IT neurons in the macaque, and initially they first identify for each IT neuron what sets of stimuli the neuron prefers or doesn't prefer. So in this case, this is a neuron that will fire strongly for dogs of small, medium, or large size, but doesn't fire much for elephants of any size at all. And the authors then show the macaque transitions of images across dogs and elephants. So here there's what they call a swap condition, where the macaque may occasionally see a medium dog followed by a large elephant, or a medium elephant followed by a large dog. But otherwise, the animals or the images earlier in the sequence remain the same. And we can see an example or a cartoon example of what they see in IT neurons throughout learning. So before exposure to this task, you have a neuron that will fire strongly for dogs and a neuron that doesn't fire strongly for elephants of any size. But after learning, you see that closer to the swap condition, the neurons will begin firing less for, let's say, these large dogs and firing more for these larger elephants. And so this is an example where somehow the transition statistics of the environment will affect visual encoding. And we can also find a similar effect in our model when we simulate this task. So here are two example neurons in our model, and we can see a similar kind of change within the neuron encoding. And importantly, we also quantify this to show that this is significant. These are just a couple of examples to give you a sense of what things look like. And so within this task, you really need a predictive module added into this network to get this full effect. And so across these two experiments I've shown you, this really argues for the need to consider interactions across multiple modules to get the full breadth of experimental results that's seen in literature. And so there was a lot of interesting aspects to this paper that I would love to talk more about. One thing that I didn't get to mention is within our module, we have both a predictive and a contrastive loss, and we have thoughts about how that could be an interesting model for pattern separation and completion within the hippocampus. The other is that you might have noticed we're not using prediction for sequence generation or forward planning, but we think this is interesting because this also suggests an often overlooked benefit of having a predictive module, which is that this system could be very useful for learning representations that are used in other areas. Also looking at things in this deep reinforcement learning setting as opposed to a tabular reinforcement learning setting really exposes new representation needs, for instance, the need for auxiliary objectives. And so this really argues for looking at things in a more complex setting. Finally, we think that this could be a very interesting normative approach to look at multi-region modeling, and that there's a lot of future directions that can be extended from this model. So with that, I'd like to thank my advisor, Kim, as well as my main PhD advisors, Larry and Dimitri, and the many labs who have given us feedback over the course of this project. If you're interested, you can find our paper on arXiv, or contact us afterwards or chat with us afterwards. Yeah, we're really excited to talk more. Thank you.
Thank you. I think I see a question on this side.
Really exciting work. I'm curious about what the reinforcement learning objective bring you. It seems like a lot of the phenomena you observe can be explained by the predictive objective. Like what happens if you train the agent just by a purely predictive objective, for example, sound, like decision transformer kind of objective?
Yeah, that's a good question. I think a lot of the results we show, there isn't one experiment, at least from a neuroscience perspective, that really gives the need for having all modules together. So it's sort of having to look at all these experiments together. Some experiments could, in fact, be explained by purely prediction and encoding, but there are other experiments that really require value learning to get that kind of effect. And you do see value learning effects both in visual cortex and in hippocampus that you wouldn't get if you ablated this value learning system. So from I guess, a neuroscience modeling perspective, it is important to have that system in there.
Thanks.
I think one question here while the next speaker comes up and sets up.
So actually, I have the same question as the previous person have a question. So instead of having, so as I see you actually combine these two laws in a linear way, actually, do you have any intuitive sense why it should be combined linearly instead of any other forms of these two separate losses?
Yeah, that's really interesting. Yeah, we kind of just started out with sort of what people in the ML literature tend to do. So a lot of this model was inspired by previous deep RL works where this is kind of how they combine losses. But yeah, I think what you're touching upon could be a cool way to extend things. So like, for instance, what if the prediction is exponentially scaled in some way? Or if we like move away from this L2 norm difference and do something that is like more nonlinear. Yeah, I think that could be a cool way to bias representations or say, focus on different aspects of the tasks that you're interested in.
Yeah, thank you. So why not, as the previous person asking the question, separate into a different task? Like one is just a predictive task and train the predicting module separately and then combine them together so that you have just one loss for each different task, like a mini task in the StarCraft training from the DeepMind, so sort of thing like that.
Yeah, here for simplicity, we just had all of the learning combined together and everything trained end to end, but there's certainly a lot of literature where, yeah, you have different modules being trained or different training speed across each modules, and yeah, I'd love to say more. I think like that's another very important thing in terms of modeling because we know that learning rates in like visual cortex and hippocampus are quite different, so from a neuroscience perspective, it is also really important to consider that. I think for sake of time, I'll leave it there. But yeah, we'd love to chat more about ways to change that. Thanks.
Thank you very much.
Our final speaker of the session is Christopher Zimmerman, who will speak on a neural mechanism for learning from delayed postingestive feedback.
All right. Thank you. I'm a postdoc at Princeton in Ilana Witten's lab. And today I'm going to tell you about a strategy that the brain uses for learning across extremely long delays. So we all know that animals learn from experience. And typically this involves updating the value of sensory cues like sights or sounds based on rewarding or aversive feedback. For example, this mouse will learn that the appearance of the blueberry bush predicts the berry's delicious taste. And this learning process unfolds over seconds. And we have a good understanding of the mechanisms that mediate credit assignment in this fast time scale. However, there's another time scale of learning at play in this photo. After eating, post-ingestive signals from the gut tell the brain whether foods were toxic or safe. These signals can update the value of those foods, even though the feedback arrives minutes or even hours after a meal is ended. And we know very little about how the brain is able to bridge this long delay to support learning. And this is a really interesting puzzle because the mechanisms that are thought to mediate credit assignment on short time scales, like on the previous slide, may not be relevant here because they generally rely on persistent or sequential representations of the stimulus that then overlap in time with the feedback signal. This would be surprising with delays of minutes or hours. We used a classic conditioned taste aversion, or CTA paradigm, to study how the brain bridges this delay to support learning. In CTA, mice are allowed to consume a tasty drink, sweetened Kool-Aid, and then 30 minutes later are given an injection of the drug lithium chloride, which triggers a visceral malaise that feels similar to food poisoning. 2 days later, we can use a preference test between the paired flavor and water to test whether learning has occurred. And our entry point in studying this one-shot learning problem is the fact that mice only learn in this paradigm if the paired flavor is completely novel to them, as shown on the left. If the same flavor is already familiar and known to be safe, there is absolutely no learning. And this suggests that flavor novelty primes animals for learning across long delays. Our goal is to use this behavioral difference between novel and familiar flavors as a window into the brain processes underlying this learning. First, we need to know where in the brain the signals for flavor novelty and visceral malaise converge, because this could be a key site where plasticity that underlies learning occurs. To accomplish this, we map neural activation across the entire brain at every stage of CTA learning, immediately after flavor consumption, and during delayed post-ingestive malaise, and days later during memory retrieval. And we did this for mice for which the flavor was completely novel, or for a separate cohorts of control mice for which the flavor was familiar. We first used light sheet imaging to generate brain-wide reconstructions of FOSS expression as a proxy for neural activation. We then used a deep learning pipeline to identify all of the activated cells in this data set and mapped these cells to the Allen Atlas. And finally, we used statistical models to specifically estimate the effect of flavor novelty on activation of each brain region at each time point during this process. And to get our bearings, we first looked at brain-wide activation during initial flavor consumption, before any malaise or learning has occurred. And in this plot, each dot represents a brain region, and the y-axis represents our estimate of how novel at the top or familiar at the bottom, flavor selective that brain region was. We first found a set of regions that during consumption were significantly activated by novel flavors that support learning. These regions included sensory areas like the insular and piriform cortices, as well as several subregions of the amygdala. And this was really reassuring for us because there's a huge literature implicating sensory and amygdala regions, especially the insula, in taste processing and conditioned taste aversion. But we were surprised to also find a set of limbic regions, especially the lateral septum and the PFC, that were more strongly activated by familiar flavors. We think that these regions play a role in blocking learning when flavors are recognized as safe, but I don't have time to talk more about that today. Together, these two sets of observations validated for us that our brain-wide imaging pipeline is both capable of recovering regions that we already know are involved in CTA, as well as discovering new regions that are engaged during this learning process. So how do malaise signals, which ultimately drive learning, alter these flavor representations? While a similar number of brain regions were activated by novel and familiar flavors during consumption, as I just showed you on the previous slide, we found that malaise triggered a brain-wide shift towards activation by novel flavors that promote learning and away from activation by familiar flavors that do not. And this shift persisted when the memory was retrieved days later. You can see this as an upward shift at the later stages on these violin plots. And this was really intriguing to us because it suggests that the condition that's driving learning, flavor novelty, may also be producing more brain-wide activation. And we wanted to know whether there's any structure to this brain-wide shift in novel flavor activation. To test this, we simply clustered brain regions with similar activation patterns in this experiment. And we were really excited to discover one cluster of regions at the top of this heat map that was preferentially activated by novel flavors that support learning at every stage of conditioned taste aversion. And this cluster primarily encompassed several subregions of the amygdala. And this was really exciting to us for two reasons. First, the fact that a novel flavor representation that was initially formed during consumption might still be present 30 minutes later suggests that this network is a site of convergence for flavor and malaise signals. Second, the fact that this representation persists when the memory is retrieved days later suggests that the amygdala might also be a site of storage and recall for this behavior. And the amygdala is also a strong candidate for bridging this long delay after a meal because we know that it directly receives visceral malaise signals from the gut via a population of cells in the hindbrain called CGRP neurons. For example, malaise signals directly activate amygdala projecting CGRP neurons. Optogenetic activation of CGRP neurons can condition a taste aversion to novel but not familiar flavors. And CGRP neurons also more strongly activate the amygdala when preceded by consumption of a novel flavor. And all of this is just like lithium chloride-induced malaise. Okay, so our brain-wide measurements suggest that the amygdala is where novel flavor and malaise signals are converging in the brain. But this leaves open the question of how these temporally separated signals are integrated at the single cell level. One hypothesis is that novel flavor coding neurons, but not other neurons, are persistently activated for long periods of time after a meal ends, and that this provides passive overlap with incoming malaise signals from CGRP neurons. Alternatively, CGRP signals may specifically reactivate novel flavor coding neurons, but not other cells. And finally, another possibility is that CGRP signals may activate a completely separate subpopulation of cells in the amygdala that subsequently become incorporated into the representation of the novel flavor during consolidation. Each of these mechanisms could explain our FOSS data and support learning, but to distinguish among them, we need to track the activity of individual neurons across the stages of learning. To do this, we developed a strategy for chronic high-density neuropixels recordings in the amygdala combined with optogenetic activation of CGRP neurons in freely behaving mice. We ran these animals through a variant of our conditioned taste aversion paradigm in which they were first allowed to drink a novel flavor or as an internal control, water, and then 30 minutes later received intermittent CGRP neuron stimulation to induce a virtual malaise state. I'll keep a reminder of the behavioral paradigm on the left and of the hypotheses on the right. We recorded around 1,000 amygdala neurons in this paradigm, and the first thing we did was classify each cell as novel flavor or water preferring, or neither during the initial consumption. We then tracked each of these groups of neurons for the remainder of the 90-minute experiment. The first thing we noticed is that after consumption ended, each group of neurons began firing much less. This is inconsistent with a credit assignment mechanism based on special persistent activity of novel flavor neurons, as in hypothesis 1, so we can cross that out. Now, what happens when we begin to activate these malaise-promoting CGRP neurons? Really remarkably, we found that activating CGRP neurons strongly and specifically reactivated the neurons that represent the recently consumed novel flavor, but not the other groups of cells in the amygdala. And this is really only consistent with the second hypothesis that I laid out, which is shown in full on the right. Zooming in on individual 3-second bouts of optogenetic stimulation, we can see that this reactivation is driven by fast input from these CGRP neurons and not by slower changes in physiological or emotional state. And population dynamics in this experiment give us another way to look at this learning process. Population activity during consumption of the novel flavor in red and water in blue were really highly distinct. Projecting the dynamics during CGRP neuron stimulation into this same subspace in green revealed a remarkably similar trajectory to the experience of consuming the novel flavor, again consistent with the hypothesis on the right. While we were intrigued to see that malaise signals could selectively reactivate the novel flavor representation, we now really wanted to know if this reactivation might serve to stabilize the flavor representation during memory recall, as our model suggests. To test this, we took advantage of the stability of our chronic recordings to track individual neurons across conditioning and retrieval days. This experiment showed us that at the population level, the amygdala's response to the novel flavor was really stable after retrieval. So we wondered if malaise signals might play a role in this stability. Dividing the novel flavor neurons based on how strongly they were activated by these malaise-promoting CGRP cells on the conditioning day revealed a really striking effect to us. The amygdala neurons with the strongest CGRP neuron input in the middle responded much more intensely to the paired flavor when the memory was retrieved days later, whereas the remaining neurons on the right had no change or a slight decrease in their responses. And this suggests to us that CGRP activation during malaise triggers plasticity in a subset of amygdala cells that serves to stabilize the population-level representation of the flavor after learning. And altogether, we think that these findings may provide a mechanism for learning from delayed post-ingestive feedback. First, our brain-wide FOSS dataset revealed that a network of amygdala regions is activated by novel flavors at every stage of conditioned taste aversion. Next, our chronic neuropixels recordings showed that post-ingestive malaise signals really specifically reactivate the amygdala's representation of a novel flavor, and that this is mediated by these hindbrain CGRP neurons. And finally, we showed that these post-ingestive reactivations induce functional plasticity in a subset of amygdala neurons that ultimately stabilizes the flavor representation following learning. And at a broader level, we think this could provide a general framework to use for learning across long delays that relies on dedicated circuitry for detecting novel or salient experiences and, crucially, built-in priors like reactivation of recently consumed novel flavors. And I really like this framework for two reasons. First, it suggests that the common phrase we associate with unexpected nausea, that it must be something I ate, is literally built into the brain in the form of this evolutionarily hardwired prior. And second, because looking beyond conditioned taste aversion, learning across long delays is a general problem that we face in the real world that many neural models of learning don't address. With that, I'd like to thank everyone who contributed to this work, especially Alana Witten, and you for your time.
All right, let's take the question here.
Yep. That was a really beautiful talk. Thank you. I'm curious to know if you were to try to do this task with, instead of a novel taste, with like a novel shape and a familiar shape. I'm presuming that this long-term, I don't know that much about the aversive learning, but I'm presuming that this long-term learning tends to be pretty specific to tastes. So the question would be, what do you think would happen? Is it the novelty that is pulling in the amygdala, or is it a novel taste specifically?
So you're correct in that this is very specific for taste. So early psychological experiments showed you cannot do this with sights or sounds or shapes when there's a long delay before malaise. How this specificity occurs, we don't have data right now, but I think it's a conjunction of novelty, signals, and taste that are tagging individual cells in the amygdala, and that is specific for taste. The reason I think that conjunction is where the specificity is that we know that these CGRP neurons project really broadly within central amygdala to 90% of the neurons or something like that. So I think the specificity arises at that earlier stage.
But so do you have data that would rule out an alternative hypothesis that basically novelty causes some fear in mice, malaise causes some fear in mice, and that drives a conjunction?
So I think I don't have hard data on this. The reason I don't think that it's novelty that can trigger, for example, learning with anything is that these CGRP neurons that project in are not novel activated, and that gives some amount of specificity to the malaise signal coming in. Whether novelty, for example, if you have a familiar flavor and present a novel sound at the same time, and that novel signal that's unrelated to the flavor is sufficient to form that association earlier, I don't know.
Okay, thanks.
Let's take one more question.
Hey, so stunning, epic experiment, and let you discovered a whole new circuit. But I suppose the one thing that's missing for me is what you were talking about earlier, which was how does the amygdala know to reactivate those neurons and not other neurons when it gets the input from the hindbrain?
Yeah, that's the big question. So our working hypothesis, which we're in the process of testing, is that there's some biochemical signal that initially tags neurons into this ensemble. You can think by analogy to Sheena Jocelyn's work looking at elevations in cyclic AMP or Krebs, and that activation persists for some long period, and that long duration of increased excitability may be what leads to those cells being more strongly reactivated. And I think something nice that falls out of that, if it's true, which I don't know, is that it allows us that biochemical signal may put a bound on the duration between stimulus and feedback signal that would facilitate learning. But that's speculation.
All right, thank you. So we now have lunch and posters too, and just as a small reminder, anyone who's interested in board games, these will be continuing throughout the poster session somewhere.